Well, good morning. I hope you've got your thinking caps on so early in the morning. It's often said that the first lecture of the day after breakfast is one of the times where our mind is the keenest, and we do need a keen mind today to follow all that's being said. So I'm hoping to speak to you this morning about the biochemistry and the neuroscience of vision. And I've worked for 10 years in an ophthalmic company, of head of research and development at a company called Saywell Pharmaceuticals. And then 10 years after that, as Andy has mentioned, I was a school teacher teaching physics, chemistry and biology at A-level. But I did actually stop last summer, so I'm not actually teaching now. But I'm also a pastor of a small independent Baptist church in Folkestone. So we've got two subjects this morning: the biochemistry of vision and the neuroscience of vision. We're going to focus first of all on the biochemistry of the eye, and of course if we're talking about the biochemistry of the eye, we're talking about the retina. And so we'll go into that. Then we'll look at the neural signals within the brain, and how the material brain serves the mind. So there's a little sidebar here so you know whereabouts we are in the lecture. So the structure and function of the eye is very much O-level biology. You can still do O-level biology, by the way. Cambridge run a course, and it's particularly popular with homeschoolers. It's a little bit more in depth than GCSE. And if you did O-level biology, all of these different parts of the eye will be known. Our focus really is at the back of the eye, and that's where the retina is, and the photo receptors, rods and cones within the retina. Now, of course, the human eye is far superior to any modern camera. And it's no surprise, therefore, that creationists have said that the eye is designed by God. Darwin himself, when that argument was put to him, saw the power in their argument. And he said that to suppose the eye could be formed by natural selection seems, I confess, absurd in the highest possible degree. Well, Michael Behe has written a famous book, Darwin's Black Box, which I do recommend to anyone to read. But he also wrote this book, Darwin Devolves. And he mentions this quote by Charles Darwin. And he goes on to explain how Darwin then moves, having said that, from scientific reason to the imaginary timeline of evolution. So we have in the eye just a few design features. We have a stretchy lens. I'm sure you know that the lens in your camera is made of glass, and it's hard and it's fixed. But we've got a stretchy lens that can be short and fat for near sight, looking at near objects, and then stretched out by these muscles which pull the lens into a different shape, and it gives very good for farsightedness. So the change in focus, as I'm sure you're aware, if you look at your finger and then you look at a distant object, is silent and almost instantaneous, a silent autofocus. The pixel density of the eye is about 50 times what you would have on if you had something like an iPhone. And of course, we don't use all of those pixels. For 576 megapixels, only about 1% is used, and that's really good for efficiency because that's all that we need. But we have a much greater receptive layer than any modern camera. And of course, the image that we see is processed by the brain, and it's smooth unstable. And I'll go on a little bit later on to speak about the image stabilization system that's done not just in the eye, but in particularly in the brain. And this is how many modern cameras work today. Of course, we have an automatic aperture. That's how wide the gap is for the light to come through. It's formed by a pigmented muscle. It gives you the beautiful colour in your eye. It's the iris. And this is made of two types of muscle. The radial dilator muscle and the radial sphincter muscle. And when there is dim light, the radial dilator muscles contract, and in bright light, the opposite happens. The radial sphincter muscles contract. And so we can automatically, under nervous control, change the aperture of the eye and therefore the size of the pupil. I wonder if you know anything about pirates, but pirates are known for wearing patches. Do you know why they wear patches? It's not because someone's gouged out an eye. It's actually because they found that the eye takes a little bit of time to accommodate to the dimmer light. And so if they're out fighting on deck and they go below deck, they want to be able to see, so they can just flip up the patch and that eye is already accommodated for the dimmer light. And you'll probably notice the opposite. When your mum wakes you up in the morning and draws back the curtains and all the light floods in and, oh, they're so bright. But 10 minutes later, our eyes have accommodated. So there is this as well, which is the switching on of the rods and cones. Well, of course, we know that photons, light is made up of little particles called photons, which act like a wave. And they pass through the pupil. The iris, of course, frames the pupil and are focused on one particular part, which is about the size of the lead, the diameter of lead in your pencil, a place called the fovea. And it's there where we see all the cones. So you've got rods. Rods are very good for periphery vision. They're not actually coloured. So if you see things by colour in your periphery vision, it's because your brain has filled in the details. You're not actually seeing it. But you've got periphery vision, which is not really in focus, and then you have that which you're focusing on, which is at the cones. So the cones right at the fovea, and that's where we focus. So we're aware of the periphery, and that's good. I'm sure any driver will want to know about that car coming up in the corner of his eye, as it were. But we have this point of focus for fine detail. And of course, we will see how, due to signal amplification, a single photon can be detected. Well, of course, atheists have fought back against this design argument for the eye, and they mock particularly the wiring. They say it's back to front. You've got all of the photoreceptors at the back of the eye, but in front, you've got all this material. You've got all these neuron cells. And they say, well, they claim that this is one of the throwbacks of evolution and bad design. And because it has to travel, the light has to travel through all of those layers before they reach the photoreceptors. Well, what can we say about it? Well, atheists mock the design of the eye. I'm sure you've heard of this person, the blind myth maker. Have you heard of this person? He wrote a famous book called The Blind Watchmaker, but he, in fact, is a myth maker. And when he talks about the eye, he really shows his ignorance of its great design. And there's another professor, Professor Daniel Dennett. He's one of the leading atheistic thinkers in neuroscience. And he said this, that no intelligent designer would put such a clumsy arrangement in a camcorder. This is just one of hundreds of accidents frozen in evolutionary history that confirms the mindlessness of the historical process. Wonderful piece of writing. But again, that's his bias. Another person, another guru of evolution, has noted the squid. And the squid, which lives in the deep oceans, doesn't have an inverted eye like we have. It has a non-inverted eye. So the photoreceptors are at the front and the wiring is at the back, as you might design a camcorder, as was said earlier. But he, having noted the squid, he said this: did the designer have greater love for squids than for humans, and thus exhibit greater care in the designing their eyes than ours? You can see the mocking spirit in which he wrote those words. But we would say, on the contrary, the inverted retina shows the wonder of God's design. Those of you who drive will know how glare reduces our ability to effectively see. Someone's got their lights on full beam, and it dazzles us. And there's all this reflected light and we can't see the road ahead. Well, that's a difficulty. And if we didn't have an inverted retina, and particularly this layer behind, we wouldn't be able to see very clearly. So visual acuity is actually greatly enhanced by the pigmented layer which absorbs the stray light. And when we think of the retina, we mustn't just think of the photoreceptors, but also of the layer just below it, which are the pigmented epithelium. And this has a very important role as well. They absorb light very effectively. And below that, there's another layer called the choroid layer. The inner choroid layer also protects the photoreceptive layer by acting as a heat sink. So photoreceptors, this is something we need to understand, they are very sensitive, not only to light, but to be damaged by that light. And they need to be protected. And so the excess light is absorbed by this pigmented layer, which absorbs it. It's got all sorts of pigments, particularly melanin. And it's so effective, in fact, that the pupil in a person's eye appears black. So there's no reflection because it absorbs it. And there's actually other pigments in there that absorb blue light, particularly. That's the most high energy wavelengths. And those who do laser surgery can't use blue lasers because we've got this pigment that absorbs the blue. And so they have to use the next best, which is the green laser. So those who are having laser surgery, that's why. So again, we protected from the excess amount of light by the pigmented layer, and the capillary-rich choroid layer acts as a heat sink because there is a lot of heat generated, particularly in the rods and cones, and we need to somehow dissipate that heat away. And so in the same way that the skin dissipates heat from the blood and from the body, so this choroid layer dissipates the heat within the retina itself. So we've got a built-in heat sink, which is wonderful. So the squid, let's think about him. He does live in deep water. And so he doesn't need to have all this protective layers from his photoreceptors. He lives in dim water. In fact, he doesn't have any cones at all, only rods. And rods tend to be much, much more sensitive, even 1,000,000 times more sensitive than cones. So the squid, you might think he's got better vision, but it's in black and white, and it's blurry, and it's not the same detail as we would say. Probably good for escaping or finding food, but not for watching television or anything like that. So they do have, as I say, many of these more sensitive rods that are needed in the dim lighting in the deep oceans. But here's the other thing. We answer evolution how? With science. Science answers evolution. And In 2007, it was discovered that the nerve cells, which were in front of those photoreceptors, actually act as a fibre optic cable, directly channeling the light to the rods and cones. And in 2014, it was found that these improved daytime vision without sacrificing the quality of nighttime vision. And from a secular science publisher, they said this is a design feature. So quite the opposite from what we would say. Now let's just think about the retina then. It's a masterpiece of design. You've got the choroid layer, which applies the oxygen and glucose, acts as a heat sink. Then you've got the pigmented epithelium, which have many roles, as we will see. And then, of course, you have this biochemical engineering. And then above that, you have the neuron layers, and we will see that this is like a built-in computer layer. We'll find out that in a moment. So you've got about 130 million rods and about 7 million cones. As I say, rods are about 1 million times more sensitive than cones. And they're responsible for seeing motion, things that move about. But they cannot perceive colour or sharp detail. And then, of course, you've got the cones. And there are 3 types, red, green and blue. They give high dynamic range colour. It's been said that 12% of women, a study was done in 2010, 12% of women have possibly an orange cone as well. And so they can see 100 million shades of colour. And that's why women are much better at decorating and knowing the very subtle shades. And my wife, it all looks white to me, but no, it's not. And all of this is providing data to the brain. So I want to talk to you now about phototransduction, because this is a very important part of the biochemistry. Photons of light come into the rods and they're converted, the light is converted into a neural impulse, into a membrane voltage. And these are amazing structures. Let's look at it in a little bit more detail. You've got, first of all, within a single rod, about 2,000 discs that get replaced every 20 or so days. And in these 2,000 discs, there are 40 million special proteins called rhodopsin. And as a result of this, there is a sequence that takes place, and a certain channel on the membrane of that rod opens up and it allows sodium ions to flow in. And we call this dark current. And then light happens and a sequence of events take place. First of all, 11 cis retinal flips from being in the cis form like this is retinal, into the trans form, all trans retinal. And when that happens, it activates another enzyme, a G protein called transducin, which activates another enzyme called phosphodiesterase, and this hydrolyzes what's keeping the gate open, cyclic GMP. And as a result of this, the channels close and it's depolarized. Let's look at this retinal. I'm sorry about the quality of the slide here, but you see that the retinal fits into the active site of this molecule called opsin. And when you've got retinal and opsin, it's called rhodopsin. And then light happens. We see this flip in shape, and as a result, the active site is changed, the protein is pushed out, and there is a conformational stage change in the opsin. And this dissociates the retinal from the opsin. It goes into the epithelium layer, and it gets restored back into the cis retinal, and that uses ATP. So you've got this wonderful recycling of the retinal between the photoreceptors and the pigmented epithelial cells below it. So in the dark, rhodopsin is inactive. And with rhodopsin being inactive, the cyclic GMP keeps the sodium channels open. And as a result of all of this, it becomes depolarized, and the neurotransmitter, right down at the bottom, you can see GLU, this is a neurotransmitter. A chemical is released, and this activates or inhibits some of what we call the bipolar cells, which are the neural cells, and therefore it's off. But when a photon arrives, everything wakes up. The rhodopsin activates the transducin, which activates the phosphodiesterase, which hydrolyzes 10 to the 5, 100,000 molecules of cyclic GMP. So you see the reason it's so complex is because each time one enzyme activates another, you've got amplification. And so 250 channels open or close as a result of one single photon, a 100,000 gain in signal. And all this happens in 1,000th of a second. So the photons lead to what we call an axon potential. And as I say, 250 channels open, and this neural signal is given off. Here is the layer. You've got the we're turning it upside down at the moment. So you've got the rod cells here. They release, in the dark, they release glutamate, which actually inhibits those intermediary neural cells called bipolar cells. They turn them off. As a result, no signal is sent to the ganglion nerve cells. They're the ones that transmit to the brain. And as a result, there is no action potential. So when the light is off, there is no action potential. But when the light is on, the glutamate is withheld, the bipolar cells are switched on, or many of them, and therefore you get an action potential. So it's beyond the scope really of this talk to give more detail. But you get edges. This is the thing, you get edges, which are very important for identifying an object and its shape and form. So just to give a little summary then of phototransduction, first of all you get activation of the rhodopsin, you get a 100,000 gain amplification. This changes the membrane voltage, and then the all-important signal is generated to the neuron. So that's phototransduction in 2 minutes. But let me move on one more thing about the retina before we move on to the brain. And that's this, that there's been a most amazing discovery in this photoreceptive neural layer, the neuron cells. You can see here that you've got the photoreceptors here, then you've got the bipolar cells, and then these are the ones that go to the optic nerve, the ganglion cells. And this is a wafer thin. Now the retina is at its thinnest 0.1 millimeter, at its thickest 0.5 millimeters thick or thin. And this actually conveys a binary code. It conveys information in binary, which is the same form that computers use. So for an image to be seen, it needs contrast, it needs edges, and that gives it shape, and you can identify it in the brain. So this built-in computer layer gives information, ones and noughts, on or off, whether the bipolar cells are on or off. And so the retina, this pre-processing of the image occurs in this retina, and the retina is like a built-in computer. So your eyes, that retina, all those neural cells act as a computer very efficiently, giving the right information that the brain needs. But let's come now to the second part of this lecture, and that's the neuroscience of vision. 95% of what we see is within the brain. The brain fills in the details, and that's the amazing thing. So I'd like to first of all think about how the neuron works, and then about the parallel processing of the visual components of that image. And then lastly, I'd just like to look at the mystery of visualization and about the mind. So there are two ends. This is a neuron. All the brain cells are neurons, and there are two ends to a neuron. They're the dendrites, so you've got the receptors for the neurotransmitters, and then you've got the other end, the axon terminal. And this connects to many other dendrites as well. And as a result, there's a flow of positive ions. And this is so wonderful. The design of the neuron itself, just a single cell, is well worthy of a Nobel Prize. Of course, the connections are all important. There are 80 to 100 billion neurons in the average human brain, and 1,000 trillion connections. So this is like every star in the Milky Way having 1,000 connections with each other. And it's the interconnection of those neurons that gives these brain activity. And all of this within a 1.2 litre, 1.4 kilogram engine we call the brain. And neurotransmitters themselves are a whole science in themselves. But let me I really need to tell you about what we call an axon potential. I'm sure you've had in the past these old cameras. We use phones today. But it used to be a little shutter button. You press it down and you keep pressing and then you get a click. And that's how a neuron works. You apply pressure until you reach a threshold level and then click, the shutter opens. It's all or nothing. So you've got these plus and minus voltages taking place. We'll just talk about that for a second. Here is a graph. You've got millivolts on this side and time. Possibly the whole of this is about 2 milliseconds. So the dendrites are stimulated or inhibited by different neurotransmitters. And this causes the voltage to go up and down, what we call graded potentials, until the threshold, and then click. The action potential fires and the signal is transmitted. Now, along the action potential, you have little proteins embedded into the membrane. And the resting membrane potential is about minus 70 millivolts. But when it reaches that threshold voltage, minus 55 millivolts, the sodium channels open and the sodium ions come through. And it becomes more positive as a result of the influx of the positive sodium ions. And it becomes what we call depolarized. But it gets to a point where no more sodium can come through. It reaches about plus 30 or plus 40 millivolts, and a special plug stops the gap, and as a result it's temporarily deactivated. And then there are other channels which we won't go into about potassium channels helping to restore the voltage, but we can see it on this graph here. You've got all of these failed initiations, and then you get to the threshold, minus 55, and click, it fires. It depolarizes, it gets to the plus 40, it closes, at plus 30, it's temporarily deactivated, and then the second half, the potassium ions flow out, restoring the potential difference and ready for the next signal. So this is a whole row of these. So along the axon, which can be up to a metre long, you've got the very first gate, and when that opens, the positive ions flow in, and the flowing in of the positive ions opens up the next gate, which opens up the next gate. And so what you have is a flow of positive charge along the axon. And this all happens at 270 miles per hour. This is what we call a neural signal. So it's not electricity, it's the flow of positive ions opening these gates. as it moves along. So the question is, if all the sodium ions are flowing in and all the potassium ions are flowing out to restore the voltage, how are you going to get back to the original concentrations? Well, there is another protein within the axon membrane, and this is a sodium-potassium pump, and it's powered by ATP. And so in the first side you've got the axon potential, the sodium ions moving in, potassium ions moving out, and then to restore it you've got this little pump which pumps them back, ready for the next signal. All of these wonderful little molecular machines that are happening in all of our neurons. Eventually it reaches the terminal, it opens up the calcium channels, it mobilizes the vesicles which contain the neurotransmitters, and then they dock onto a protein and are released. Now, of course, within the neuron, there are no ribosomes in the axon terminal. So all of the neurotransmitters, many of which are like proteins, small proteins, they need to be made in the cell body. They get packaged in the endoplasmic reticulum, into vesicles, and then get transported along the axon by these wonderful motor proteins, which are my all-time favourite proteins, kinesin. And kinesin is an amazing walking molecule. It takes a long journey along the axon, up to about 1 metre. It travels at 1 centimetre per hour, which is very fast. On a molecular level, it's about 80 steps every second. And that brings the neurotransmitters to the axon terminal, where they're needed for the transmission from one to the other. So I've got no time to talk about the various kinds of neurotransmitters. They register pain, they relieve it, they act on muscles. Psychoactive drugs are all part of this area of neurotransmitters. And they have, of course, a whole mechanism for the reuptake of these of the neurotransmitters themselves. So these are wonderful structures. But I'd like to just lastly, or thirdly, consider the processing in the brain. And I'd like to first of all talk about pathways of neurons and then the processing areas within the brain. There are major and minor visual pathways. The minor, which takes into the periphery rods, they go to the brainstem, they go to a relay station, into the visual cortex, right at the back of the brain, and then, of course, the motion is stabilized. So we've got this image stabilization system taking place in the brain. So the thalamus, which is at the center of the brain, it coordinates all this information, the brightness, the movement, the body position from other parts of the body, and it shifts the focus as a result. And all this feedback goes to the eye, and it makes these minor adjustments. So this is like processing of the image, stabilizing the image. But we're going to consider the primary pathway. It goes through the thalamus into this particular part of the thalamus, we just call it LGN, to the visual cortex. I've got a life-size model of the brain here. This is just half a brain. The other half is somewhere about, who knows. But the visual cortex right down here at the back, and you can see the different parts of the visual cortex there. And this is the main processing area. It's a bit like the sorting office of the post office. So all this information comes in, all these letters, and they need to be sorted out by different postcodes, and it's a sorting station. That's what the visual cortex does. It sorts out the information. Each channel from the eye is conserved. It projects to the visual cortex, goes down 2 pathways. I won't go into them now, and it eventually reaches the visual cortex. What happens next? Well, the visual cortex itself is only 2 millimeters thin, and it separates the components of vision. It's what we call multi-core processing, and we hear this in technology today. Modern technology is on the catch-up. But just look at it very briefly. This is a visual cortex at the back of the brain. You've got the area V1. This looks at orientation of the object and the direction that it's pointing and moving. Then you've got another area which looks at lines and edges. and that's very important in regarding shape, giving information regarding shape. And then another area analyses the angles and the depth perception. There are two outputs, pathways from the visual cortex. The dorsal route, going upwards, this has to do with action to the parietal lobe at the top of the brain. So you can take immediate action, and then you've got the ventral pathway, which is more to do with recognition. So the ventral route you've got from visual area 4, this really rich in the neurons that come from the fovea concerning colour and fine detail. It goes to the temporal lobe, where it connects to a database in the brain. This is why I'm saying that we see only about a few % of what the image that's formed in the brain. And this database pulls out all this information once that object is recognised. It fills in the missing parts, including memories. So faces and words then begin to take on meaning, and we're beginning to understand what we're seeing. So this is all wonderful taking place on the bottom part, the inferior temporal lobe down here. And then of course you've got, so as a result of this, the visual stimulus becomes what we call conscious perception. Then the dorsal route starts at V5, just near the temporal lobe. It goes to the parietal lobe that analyses action. Someone throws something at you, do I need to duck? You need to know. Often you duck before you even recognise what it is. And that's because it's a faster processing. It goes to the dorsal route. And of course, it also, in projecting to the parietal lobe, coordinates not only the quick response, but it combines learned behaviour, which is very important for sporting activity. So it's a tennis ball, and you know how to respond to the object coming in. So here's a picture of a sequence of any action. Requires a coordination of the brain regions, the focus, integrating the senses and memory, creates a plan of action, and then it projects to the motor cortex for an output signal. So you first assess the situation. This is conscious thought, goes to the frontal part of the cortex, the frontal lobe, and that's for conscious thought. Then get set. The learned actions are called upon in the brain from the putamen, and we're ready for action. And then you have coordinated action is planned. It stimulates the premotor cortex, which is just the base here of the brain. And as a result of this, you've got the motor cortex project to all the appropriate muscles, the eyes, the hands, the arms, the legs, wonderfully coordinated within the brain. And you've got fine control feedback from the very base of the brain, the cerebellum, and that gives you the fine feedback. So we spoke a little bit about the motor cortex. Some responses are made by the hypothalamus, another part of the brain. That includes things like the changing of the iris, opening and closing, the focus, the ciliary muscles, blinking, all that kind of thing. But most of the voluntary movement comes from the motor cortex, under the watchful eye of the cerebellum. So that you get this constant feedback and regulation. So in summary of the brain, the components of vision are separated in the visual cortex. Motion is analysed in the parietal cortex. It's recognised in the inferior temporal lobes. The frontal lobes assess the situation, feeds back. Eventually the motor cortex projects to the muscles and is regulated by the cerebellum. Wonderful coordination of all these different processing areas within the brain. But in conclusion, I'd like just to speak about the mind. How do we visualize? Is it in the brain where there's no more than neural activity, or is there something we have called the mind? Well, neuroscientists are divided when it comes to the whole subject of visualization. Most are secular, and they say all of visualization, all of consciousness and perception is within the brain. It's like the sum of the parts, like an orchestra. That's their best understanding. I won't read all of this, but they know how the brain works, about visual perception and all the different parts. I'll just skip through that. But they do admit that there are still many questions to answer. And here it says, perhaps our perception of specific objects is based on the excitation of a small number of specialized neurons in some ultimate perception area that has not yet been identified. So here is the end of neuroscience. They don't yet know how we see things. They can only say we can know what happens in different parts of the brain. So is thinking about what we see exclusively material or neural activity, or do we have a mind? Now, to monists, the mind is the brain. And atheists like Dennett are very strict monists. But there are also dualists, the inspired writers of the Bible. The Christian worldview is that the mind is separate from the body. Descartes, very ahead of his time, said there are great differences between the body and mind. We won't agree with all his conclusions, but he was 400 years ahead of the current debate. So I would say that dualists believe that the mind is not matter. The fundamental truth is that we have both a body and a mind. That's the biblical view of man. We have both body and soul, brain and mind. And we freely admit that there is a close relationship between the brain and the mind. But conscious thought is very much in the mind. We visualize in the mind. We would hold this, that the brain is the servant to the mind. It's with the mind that we worship our Creator. It's the arena in which we reason and have faith and conscious decision making. Consciousness is in the mind, which is separate from the brain. And we will continue, and this is why it's important, we will continue to have consciousness after death. Consciousness is still a mystery, but the Bible is unshakably clear that consciousness continues after death. So the mind, therefore, cannot be synonymous with the brain. One certainty we all agree is that we are fearfully and wonderfully made. God has created both how the eye sees and how the brain should work, and we should give him all the glory. Thank you, Andy. Sorry I've gone a little over time.